What is mitosis? Mitosis is a type of cell division in which one cell divides to produce two new cells that are genetically identical to itself. In the context of the cell cycle, mitosis is the part of the division process in which the DNA of the cell's nucleus is split into two equal sets of chromosomes. The goal of mitosis is to make sure that each daughter cell gets a perfect, full set of chromosomes. Cells with too few or too many chromosomes usually don't function well. They may not survive, or they may even cause cancer. So, when cells undergo mitosis, they don't just divide their DNA at random and toss it into piles for the two daughter cells. Instead, they split up their duplicated chromosomes in a carefully organized series of steps. In the early prophase, the cell starts to break down some structures and build others up, setting the stage for division of the chromosomes. In metaphase, the spindle has captured all the chromosomes and lined them up at the middle of the cell, ready to divide. Before proceeding to anaphase, the cell will check to make sure that all the chromosomes are at the metaphase plate, with their kinetochores correctly attached to microtubules. This is called the spindle checkpoint and helps ensure that the sister chromatids will split evenly between the two daughter cells when they separate in the next step. If a chromosome is not properly aligned or attached, the cell will halt division until the problem is fixed. In an anaphase, the sister chromatids separate from each other and are pulled towards opposite ends of the cell. Lastly, in telophase, the cell is nearly done dividing, and it starts to re-establish its normal structure as a cytokinesis, or division of the cell contents, takes place. Mitosis is used for almost all of your body's cell division needs. Meiosis, on the other hand, is used for just one purpose in the human body: the production of gametes, sex cells, or sperm and eggs. Its goal is to make daughter cells with exactly half as many chromosomes as the starting cell. To put that another way, meiosis in humans is a division process that takes up from a diploid cell, one with two sets of chromosomes, to haploid cells, once with a single set of chromosomes. In humans, the haploid cells made in meiosis are sperm and eggs. When a sperm and an egg join in fertilization, the two haploid sets of chromosomes form a complete diploid set, a new genome. In meiosis, however, the cell has a more complex task. It still needs to separate sister chromatids, as in mitosis, but it must also separate homologous chromosomes, the similar but non-identical chromosome pairs an organism receives from its two parents. These goals are accomplished in meiosis using a two-step division process. Homologo pairs separate during a first round of cell division called meiosis 1. Sister chromatids separate during a second round called meiosis 2. Since cell division occurs twice during meiosis, one starting cell can produce 4 gametes. In each round of division, cells go through 4 stages: prophase, metaphase, anaphase, and telophase. During prophase 1, differences from mitosis begin to appear. As in mitosis, the chromosomes begin to condense, but in meiosis 1, they also pair up. Each chromosome carefully aligns with its homologo partner so that the two match up at corresponding positions along their full length. After crossing over, the spindle begins to capture chromosomes and move them towards the center of the cell, the metaphase plate. This may seem familiar from mitosis, but there is a twist. Each chromosome attaches to microtubules from just one pole of the spindle, and the two homologous of a pair bind to microtubules from opposite poles. So, during metaphase 1, homologo pairs, not individual chromosomes, line up at the metaphase plate for separation. In anaphase 1, the homologous are pulled apart and move apart to opposite ends of the cell. The sister chromatids of each chromosome, however, remain attached to one another and don't come apart. Finally, in telophase 1, the chromosomes arrive at opposite poles of the cell. In some organisms, the nuclear membrane reforms and their chromosomes decondense, although in others, this step is skipped. Since cells will soon go through another round of division, meiosis II, cytokinesis usually occurs at the same time as telophase I, forming two haploid daughter cells. Cells move from meiosis I to meiosis II without copying their DNA. Meiosis II is a shorter and simpler process than meiosis I, and you may find it helpful to think of meiosis II as mitosis for haploid cells. The cells that enter meiosis II are the ones made in meiosis I. These cells are haploid, have just one chromosome from each homologous pair, but their chromosomes still consist of two sister chromatids. In meiosis II, the sister chromatids separate, making haploid cells with non-duplicated chromosomes. During prophase II, chromosomes condense and the nuclear envelope breaks down if needed. The centromes move apart, the spindle forms between them, and the spindle microtubules begin to capture chromosomes. Lastly, in telophase II, nuclear membranes form around each set of chromosomes, and the chromosomes decondense. Cytokinesis splits the chromosome sets into new cells, forming the final products of meiosis. 4 haploid cells in which each chromosome has just one chromatid. In humans, the products of meiosis are sperms and egg cells.